Okay, so next up we have Elizabeth de Guzman, who will be presenting some work that she conducted when she was in Alessandro Gozzi's lab on some optogenetics.
There we go. He's a lot taller than me. Thank you very much for the introduction, and I have nothing to disclose. So the topic that we're interested in this session has already been addressed, but we'll come back to it just briefly. What we mean by functional connectivity, as you're probably all familiar with, it's a metric defined by the temporal correlations between spontaneously fluctuating signal across the brain. In our case, measured with fMRI, and for my talk, I'm going to be talking about bold signal again. So there's a lot of agreement in the community that the functional networks we can map in this way are pertinent for understanding brain function due to their association with cognitive processes and frequent dysregulation in human brain disorders, but there remains a lot of open questions in the field, in particular what properties of the underlying neural activity are reflected in this metric. And you're going to hear about a lot of different ways during this session about how to address this question. But some of the techniques that have been done, particularly in humans, is relying on multi-modality recordings and using correlations to try and understand, in particular, what neural rhythms play a role in functional connectivity. So I'm showing here just some examples from various modalities, ECOG, intracranial EEG, and MEG, which have tried to identify the power in each electrophysiological frequency band that co-varies best with the functional network mapping. And so you can see, at least from this subset, there's maybe sort of a bit of overlap in the different frequency bands that these studies find, but there's not sort of full consensus within the field. And there's one big problem that these studies can't get around, and there we go. And that's the fact that these frequencies are all intertwined. So they're nested within each other, and their powers co-vary. What I'm showing here is an example from MEG recordings, and this is just showing, like, each panel is a different region in the brain and showing how power across different frequencies are locked to the phase of the slow oscillation. So you can see how it's difficult to separate the individual contribution of these different rhythms just with correlation alone. So the question that we had was whether or not we could leverage causal manipulations in an accessible species, like the mouse, where we can induce different rhythms separately in order to try and disentangle these dependencies. Okay, so to do this, we wanted to modulate a network with translational relevance. The default mode network is a functional network of the human brain, widely studied. And from functional connectivity analyses, an analogous system in rodents has been identified. So to modulate this network, I targeted a hub region of the DMN. Sorry, I'm really struggling to move these slides. The medial prefrontal cortex in the mouse, and I used optogenetics to stimulate excitatory CAMK2 neurons. Now, in order to stimulate this region, we turned to a collaborator of ours, Ferruccio Pisanello, and his team at the Center for Biomolecular Nanotechnologies at IIT, and he helped us devise a method for performing bilateral stimulation of the entirety of the prefrontal cortex using optogenetics with minimal damage to the surrounding tissue. Now, I'm not going to go into the details of this technology, but what I am going to do is show you the results of a sanity check. So you know that we have the mouse structural connectome, and that's quite well defined, and that's what I'm showing here. This is the connectivity of the prefrontal cortex, the region we're stimulating, and shown in brown are all the regions that the prefrontal cortex projects to. So traditionally, fMRI studies with optogenetics classically use a block design stimulation, where the laser is pulsed rapidly in 10-second blocks, followed by 50 seconds of rest. And this causes increases in the bold signal timed to the light, sort of these blips that I'm showing here in the stimulated region. And this type of pattern is standard because it gives you the ability to probe the projections of the neurons being stimulated. So if we map all the regions of the brain where there's a significant increase in the bold signal at this timing, what we get is this map on the right here. And you can see that it overlaps quite well with our axonal connectivity. Okay, so satisfied that we could stimulate the prefrontal cortex and all of its projections, we next asked ourselves what sort of stimulation pattern could be used to address our research question. So I told you at the beginning that the powers of different frequency bands co-vary, and we want to try and disentangle this. So to do this, we chose to stimulate with nested frequencies. What does this mean? What we have is a fast carrier frequency that the laser is being pulsed at, and the amplitude of this is modulated by an envelope, a slow envelope. And so in this way, we could test separately both the effect of the carrier frequency and the effect of the slow envelope frequency independently. And to start, I'm going to show you the results of a carrier frequency at 20 hertz, an envelope frequency at 0.1 hertz, and this choice was just based on some seminal MEG work, but then we're going to expand on that. And before I continue, I just want to emphasize that before we proceeded with the experiments, we confirmed with in vivo electrophysiology the presence of oscillations in the LFP timed to our rhythmic stimulation, confirming that we could induce these slow oscillations with the paradigm that we chose. Okay, so satisfied that we could entrain our rhythm of interest, we began by mapping functional connectivity, as is traditionally done. And this is what I'm showing here for a seed region in the area that we've stimulated. And so you can see regions such as the thalamus have fairly high functional connectivity, but in the striatum, functional connectivity is 0. And if you're familiar with the mouse brain, you're familiar with the prefrontal cortex, this may be of a surprise to you. What I showed you before, when we did this block design stimulation, we actually got a really strong response in the striatum because it is a region that the prefrontal cortex projects to. So initially, this was a bit surprising to us that these two things seem to be showing disparate results. But if you're familiar with functional connectivity, you know it's a correlation. It's a fairly simple metric. There's more that we can dive into here. So I'm going to show you the results of still a very simple metric, but phase locking. And so the way that this is different is if we take our bold signal, instead of doing the correlation to get our functional connectivity, we take that signal, filter it to our frequencies of interest, take the Hilbert transform to get instantaneous phase, and then look at the difference of that phase. We can get a metric for the concentration of the phase locking. So over the total time period that we stimulated, how much of that time our two signals were locked to the same phase, and then also what that angle is. So now going back to our results, we can look at the effects in a new light, looking again at phase differences between our signals. And if we look at the phase locking concentration, we can actually see that there's an increase in phase locking everywhere, even regions where we didn't see strong functional connectivity. And so this shows that there's strong entrainment to this slow envelope frequency when we stimulate. So what's going on with our functional connectivity? Well, I'm showing you a video here. The clock shows sort of the whole period of the stimulation, and you can see the progression of sort of peaks and troughs of the bold signal throughout this. And you can see how the signal is progressing and why we might get functional connectivity that is so low. And the reason is that the signal is out of phase. So if we look in the striatum here, we can see that for for the rhythm that we're inducing, it's completely out of phase, which is what is giving us this difference. So another thing that I want to point out here is that propagation of this rhythm seems to not simply be explained by the connectome, because the striatum is directly connected to the PFC and is quite close. So before exploring this phenomenon, we wanted to verify that what we were seeing was neurally driven. We've started this talk by wanting to know the neural underpinnings of functional connectivity. So identified 3 regions with different phase locking angles and performed in vivo electrophysiology. And what I found is there were phase differences that were consistent between the 2 modalities. And this suggests a neural origin of this effect. So, satisfied that the effect we're looking at had a neural origin, the first thing that we wanted to ask was, if we changed the carrier frequency, how does this affect our functional connectivity measurements? So to do this, I mean, one of the ways you could do this is just take our 20 hertz carrier frequency and then try and sample a whole bunch of different frequencies. We decided to do something a little bit easier than that. And essentially what we did is, instead of fixing that carrier frequency at a single 20 hertz, we just introduced delays that were randomly distributed, therefore all equally likely, and then redid the study. And in fact, when we did that, we got the very same effect. And this verified, at least with this paradigm, that the carrier frequency seemed to have minimal effect on our results. So then we wanted to ask, if it's the slow frequency we're interested in, how slow is slow? What happens if we change this to a faster 4 hertz? And we did that. We looked at functional connectivity, and we saw no functional connectivity with 4 hertz stimulation. And some of you who do bold imaging may know that maybe this is because we sample only at 1 hertz. And so we teamed up with Noam Shemesh from the Champalimont. We increased the sampling frequency to 13 hertz, and we found that, no, even if we sample even faster, we still don't get any entrainment with the bold signal to our faster frequency. And just to make sure that this wasn't because we weren't able to induce this frequency, we confirmed again with in vivo electrophysiology that we could actually get entrainment. So, if it's the envelope frequency we're interested in, and if we walk that envelope frequency higher, our effect goes away. We wanted to explore what lower frequencies we could change this to and whether or not we could modulate this effect. And so what I'm showing here are the results of 3 different frequencies: 0.05 hertz, 0.1 hertz, and 0.2 hertz. This is the slow envelope frequency we're trying to entrain at. And what you can see if we look at the phase locking angle is that we get different architectures in our phase locking patterns with structure-specific differences in the response to different frequencies. And just very quickly, I'm going to point out that it doesn't look to be simply due to some sort of time delay, because when we look at different structures, we get different phase offsets, some of which are sort of bimodal in response, and some of which are like linearly increasing with increasing frequency. The last thing that we were interested in is going beyond phase. So we've been talking, I've been focusing on phase differences, but you may also be wondering, what about the amplitude of the response? And is that going to have any effect on our functional connectivity measurements? And so what we wanted to look for, since we were sampling different frequencies, was whether or not we could identify a resonance response. That is an identification of a frequency for which the response to stimulation was greatest. And so this is how we do this. I'm showing spectral density for 2 different stimulation frequencies, 0.05 and 0.2 on the bottom. And we simply computed the relative power of the stimulation frequency compared to some frequencies just outside of that. And we did that for the different stimulation frequencies. And then we looked across the brain. And And in fact, what we found is that structures heavily innervated by the PFC had a disproportionately larger response to a particular frequency compared to the other two. And I'm showing here example from the midbrain and example from the PFC showing different resonant frequencies. And this is interesting in general for fMRI coupling, but also started to reveal an important mechanism where brain regions could facilitate targeted information transfer and potentially maximize the efficiency of large-scale brain communication. So, to wrap things up, let's go back to the original goal of this symposium. What have we learned about the neural underpinnings of functional connectivity? Well, with this work, we've put into question some of the previous assumptions about the rhythms that could support and contribute to functional connectivity. I've shown that slow rhythms support functional connectivity and not faster rhythms, such as 4 Hertz. I've also demonstrated it was modulating the envelope, not the carrier frequency, that modulated the response. And we've seen that a region with low functional connectivity, a low correlation, actually had really strong coupling to our frequency of interest. And so ultimately, phase differences between our signals and how they propagate are going to affect the functional connectivity. And finally, we've shown evidence of frequency-dependent communication across networks, and this may even have implications beyond functional connectivity. So with that, I would like to, in particular, thank Alessandro Gozzi, who I did all of this work with at the Italian Institute of Technology, the collaborators who helped with some of the technology development, our funding, and all of you for listening.
We have time for questions.
Is this on? Brilliant systematic work. I think that was just amazing results. When I see the propagation, have you looked at how that relates to quasi-periodic patterns from the Kielholz lab? Is that, I'm just, making that connection. It seems similar.
Yeah, this is something we're interested in looking at, and we've talked to Shella about this. One thing I'm not sure I emphasize so much is that these are the results of like constant stimulation. So we stimulate for 15 minutes, and we actually sort of chop off some of the data at the beginning. So we've reached some sort of steady state. And so looking at this phase analysis, it's a very simple metric, but when you're looking at only one frequency and something that's so stable, this ended up being sort of enough of analysis to dive into it, but absolutely would be interesting.
Hello, thank you very much for your talk. One thing that you pointed out, and that's something that has bothered human researchers for a while, is the fact that we use correlation analysis, which is quite simple, and you have shown that correlation fails to find important connections in the brain. How do you think your work informs human researchers into better ways to analyze our data to find functional connectivity?
Better ways. I would probably say that the work that I've done in the mouse sort of gives me the ability to control a lot of aspects of this. There are some things that could be done in humans as well. Non-invasive stimulation exists, and there's lots of ways to try and modulate sort of ongoing endogenous oscillations in the brain in humans. And so I think the thing that has to be done next, this was inducing an oscillation and just sort of overriding the system. The next step, and this is something that could also sort of be done in humans as well, would be to try and be more subtle and play with increasing the power of different endogenous frequencies to see if some of the things that I've shown still hold up under that case.